Okay, hello and welcome to our Ask a Physicist. Before we get started, I will make my usual announcements, and the first one being that our next Ask a Physicist will be April 24th at the usual time, which is 6 p.m. Mountain Standard Time, and I will send the announcements out over email and social media like usual, so look out for that for registration. And then lastly, this is being recorded as usual, so it will be posted if you want to rewatch it or recommend it to somebody you think would be interested. And if you have any questions at all, you can ask them at any point during the presentation using the Q&A feature at the bottom of your screen, and we will get to questions at the end. So with that, I will turn it over to our moderator for the evening, Maulik Parikh.
Hello, everyone. Welcome and good evening. So today's topic is a very exciting one that we all love to speculate about. Is there life outside Earth? And more specifically, is there life in our solar system? So that would be very exciting, because even if they're not aliens coming on spaceships, even if they're microorganisms, it would transform the way we think about Earth. How special we are. And we're very happy to have our first speaker, Kirt Robinson. Professor Robinson is an assistant research scientist in the Beyond Center at ASU, and he's a professional affiliate on NASA's Europa Clipper mission that will launch a spacecraft next year, near the end of 2024, to assess the habitability of Jupiter's icy moon, Europa, which has a subsurface ocean. His work is focused mostly on chemistry and natural systems, chemical mixtures that microorganisms can use to harvest energy, and how life might get started from non-living chemistry in the first place. How do we go from just molecules to life? And today is going to be talking about the possibility of life elsewhere in the solar system. And so with that, I'll pass it on to him. But before I do that, let me just say that if you have any questions, please, as usual, feel free to enter them into the chat, and we'll get to them after our two speakers today. All right, over to you, Kirt.
All right, thanks so much for that introduction. Really happy to be here. Thanks for having me, and thanks everybody for showing up. I'm going to try to load my slides here. There they go. And in that intro, it might have become apparent that I'm kind of an infiltrator. This is Ask a Physicist, and I'm a chemist. So I'm going to hope to give you a little bit of a different perspective here. Now, on my title slide, you'll see that I have a big old picture of the Earth, even though this is called Life in the Solar System. Well, the reason for that is this is our only data point for life in the whole universe. Okay, so everything we know about life comes from what we know here on Earth. So I'm going to spend the beginning of this talk talking about the limits and the requirements of the life as we know it on Earth, because we need to understand that before we can think about how we might go looking elsewhere for it. And where we're going to be looking elsewhere in this talk is Mars, Jupiter's moon, Europa, and Saturn's moon, Enceladus. And hopefully you can see my cursor. Somebody give me a thumbs up if they can. Yeah. Good. All right. So before we get into the nitty-gritty science, I want to ask a few thought-provoking and hopefully inspiring questions. The first is, if we were to discover alien life, a second origin of life, how would that change our society? Okay, is this going to be like a scientific revolution like we've seen in the past? I certainly think it would be in the field of biology. But I think it's worth mentioning, with a discovery like this, I don't think it's just going to be a party in the scientific community. I think it's really going to permeate through our entire culture, into the classroom. You know, I'd be surprised if primary school students didn't know about this event. I'd be surprised if our curriculums didn't change to incorporate this into what students are learning at a very early age. Again, definitely in the biology textbooks, chapter 1, I would think. Next, could this affect human spirituality, how we view our relationship to the rest of the universe, how we might derive meaning from life? And could this affect religious institutions? Could new doctrines be birthed from this discovery? Now, I'm willing to put money on the fact that we would definitely develop some powerful new technologies if we were to discover a second origin of life, especially if we were able to get a sample of that life back to our labs. And I think if we discovered it, we would work very hard to do so. Now, a popular example of when we go discovering new life forms on Earth and how much we can learn from that, a powerful example is the discovery of penicillin, just from curious scientists probing the mechanisms by which fungi and mold work. That was a huge discovery for the human race. I want to relate to you another example here where our technology was boosted forward as we discovered new life forms. In the 60s, explorers went to Yellowstone National Park and explored life in these hot springs, very alien to us at the time. And they discovered a single-celled organism that had a heat-resistant enzyme known as polymerase. And what polymerase does is it aids in the replication of DNA. And the discovery of this enzyme from this microbe allowed a new technology to be born called polymerase chain reaction, or PCR. This has had huge implications in the fields of medicine, agriculture, forensic science. You know, if the police find a weapon and there's a little bit of genetic material on that, it's not much good to us. We really have to replicate it a whole bunch, amplify the signal so that we can match that DNA sequence to a suspect. And we've learned all sorts of things about how humans on the planet are related to each other and their histories through time and their migrations from this polymerase chain reaction, which allows us to sequence DNA. We've also learned so much about our relationships to other species, how evolution has progressed over the course of Earth history to get from single-celled organisms to humans now. So all this just from stumbling around looking for new life forms. And I imagine that if this were alien life, we would probably learn even more and maybe even develop more exciting technologies. Okay, so let's quickly shift gears. I want to quickly share some optimistic thoughts related to the search for life in the solar system. The first thought is this: when we look around on Earth, in environments that to humans seem quite extreme, we seem to find life. At least when we're looking on the surface, and as far as we've looked into the subsurface so far, there is an upper temperature threshold. But back to these hot springs in Yellowstone, this really exemplifies the point. You can find boiling acid springs where microorganisms are just thriving. You can go to other springs that have really concentrated chemicals that we would consider extremely toxic to us. Organisms are turning them into their food. Okay, there are geysers where organisms in these outflow channels, let's say these orange regions, where it's going hot, cold, because the guys are burping out hot fluid, and then it doesn't burp it out for a while, and the organism gets cold. These massive temperature ranges would seem very detrimental to life, but there are certain organisms that just find a way. So this versatility of life makes me very optimistic that the harsh conditions we perceive in space environments and planetary environments could still be inhabited. So here's another place. Let's go to colder temperatures. This is a glacier in Antarctica, and it's belching out this red fluid. This is essentially waste from a subglacier ecosystem where microorganisms are happily chewing on the chemical mixture there to get their energy. And this is an example of sort of the opposite end, the lower temperature threshold of life. Now, we have identified an upper temperature threshold. Of course, it keeps getting pushed up with new discoveries, but we really haven't established a lower temperature threshold for life. These organisms are something around -7 degrees Celsius or about 20 degrees Fahrenheit. But really, there doesn't seem to be a lower limit so far. The real limit is, when does water freeze? Because the ice crystals damage the cells, and if it's a solid block of water, the chemistry of life can't really operate there. So if you can depress the freezing point of water with salty water or other antifreezes that sometimes organisms produce, you could potentially get much, much colder liquid where cells can thrive. And that might be important for looking around the solar system. There are a lot of cold places out there we want to look. Okay, and lastly, I want to take you to the bottom of the ocean. The extreme condition here is the pressure. So these organisms are experiencing the weight of a few kilometers of water on top of them. And again, they're doing just fine. There's a whole ecosystem here. What you're seeing in this black kind of smoky looking stuff is seawater that's permeated through the sea floor. It's gotten near a magma chamber, and its chemistry has changed as it's heated up. And then it's getting very hot and becoming buoyant again and coming out as a very different fluid and mixing with the seawater. And there's all sorts of chemistry happening. That's why it looks so strange. But what's happening in this ecosystem is these organisms, there's no light down here. This is very deep in the ocean. They're chewing on this chemical mixture of these two fluids mixing. And so single-celled organisms are sort of the primary producers, and then animals come and feast on those. So you get this whole beautiful ecosystem, kind of like a rainforest at the bottom of the ocean, where we used to not know there was any life at all or have any reason to think there was life. So maybe we'll make discoveries like these around the solar system. So again, this is why I'm optimistic. Life is very versatile. These harsh environments in the solar system could still be inhabited. Here's just sort of a summary slide. You don't have to really focus on it too much, but it's just, yes, we found life in a lot of crazy places on Earth that seem harsh. The further we look, we keep finding it. So let's keep looking and let's keep pushing those limits. Another thing that makes me optimistic is that life seemed to have gotten started on Earth fairly quickly. Now, quick is sort of a relative term when you're talking about planetary history, but when the Earth formed, it had a molten surface. But not long after that surface solidified into rock and oceans could form, we see from ancient rocks that life took hold. So if you want to approximate, we could say something like it took about 10% of the Earth's lifetime for life to emerge. That's good news, because if it only takes that long, that's about 10% of the age of the solar system. All these other planets and moons in the solar system have a lot of additional time to get that recipe right for life to emerge. And the last point I want to make about why I'm optimistic is that we living things seem to be made of fairly ordinary material. Building blocks, our basic components, are found in space rocks that are representative of the early solar system, and material that's distributed throughout the solar system and has been raining down on planets and moons for the history of the solar system. One example here that just popped up is this chnops, the elements that seem to be dominant in life. So that stands for carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. They're plentiful in these materials relative to Earth. So we don't need to worry about having the basic ingredients. And you can actually take this a step farther. Not only do we have the elemental ingredients, but a lot of the time they're actually in the right form that life uses, the right chemical form. I'll show you what I mean by this. So here's a picture of a protein. This is actually that enzyme, that polymerase enzyme. This is its 3D structure. And it's made out of sort of, you could think of it like a string of pearls. These pearls, each pearl is an amino acid. That's a type of chemical. And they're strung together. And when you string them together, because of their chemistry, their chemical behavior, they fold and they form these 3D structures that have really powerful functions in biochemistry. And it turns out when we take a look in these space rocks, we find these amino acids everywhere. We find loads of them in meteorites, so space rocks that have crashed to Earth, asteroids, which maybe represent, in many cases, the parent bodies that those meteorites came from before they crashed to Earth. So there was a recent detection in the Ryugu asteroid, which a spacecraft went, grabbed a chunk of it, and brought it back to Earth. So that's a very pristine sample. We found amino acids in comets, and we find them in simulation experiments. And by that, I mean experiments that are meant to, you kind of set up the chemistry to mimic the early Earth or mimic a planetary environment or mimic an asteroidal environment. We also can generate amino acids. And in some cases, we've actually found some amino acids strung together, looking like sort of the beginnings of a protein. I'm going to go through the rest of these building blocks kind of quickly in the interest of time, because I know we all want to get to the outer, or to our Earth neighbors, planets and moons. So we've also found little bits and pieces of other building blocks. Bits of DNA and RNA we find throughout these space rocks and experiments. Okay, I'm just going to cycle through these. We also find molecules that look like they could be pieces of our cell membranes distributed in these space rocks. Okay, so that's where all the optimism comes from. Now we need to think about what else does Earth life need? Because if we can figure out what it needs, then we can figure out the environments that are most favorable to host it. We've established liquid water is important. We've established that you got to have the ingredients, you got to have the stuff to build yourself out of if you're life. And one thing we haven't talked about is an energy source, and this is really, really important. And this is especially the chemistry perspective I want to bring to you all. So, like a car needs to burn fuel to drive around, to do its work, cells need to burn fuel to maintain themselves, to perform functions, to eat, to build their cellular materials. They need to burn fuel. So we need to think about energy sources. And I want to go to a familiar example. I want to go to a familiar example after we talk about this energy pyramid. So a lot of us are taught this in grade school, this photosynthesis-based energy pyramid. And it makes a lot of sense when you are a human that lives at the surface of the Earth, that the sunlight powers this whole ecosystem, we're taught. Sun shines on the primary producers, it gets eaten by consumers, up and up the pyramid. If the sunlight goes away, you'll notice this energy pyramid collapses, okay? And when we go looking elsewhere in the solar system, these environments are not very favorable to photosynthesis. They're essentially exposed to the vacuum of space, okay? There's not much, if any, liquid water on the surface of these things to host things that do photosynthesis. And they're bathed in harsh radiation, which is not going to make the surface a very pleasant place to live. So we need to think about a different kind of energy pyramid. And so I'm bringing back this picture of that mound on the bottom of the sea floor. And this is a chemosynthesis-based energy pyramid, okay? There is no sunlight reaching this ecosystem, but here it is. It doesn't fall apart without the sun. These chemical mixtures that are occurring when the water goes down, reacts with some rocks, and gets volatiles injected into it from the magma chamber and comes back out. That chemical mixture provides energy for the ecosystem. And now I'm going to show you the example, a familiar example, so we can wrap our heads around this. So we're all familiar with human respiration, breathing. We breathe in oxygen, or O2. We breathe out carbon dioxide, or CO2. So what's actually happening there from a chemistry perspective? Some of you may have seen this chemical reaction in primary school biology or somewhere else. This is a reaction of glucose plus oxygen. That's what we're doing in our bodies. We're just burning glucose. Those are reacting to produce CO2 and water. So we breathe out CO2 and water vapor. And at the same time, our cells harvest chemical energy to do all the things we do in our daily lives. Now, what's important to note here is that to take advantage of chemical energy, you need what's called a reductant and an oxidant. Well, oxidant's easy. It sounds like oxygen. And oxygen is a very powerful oxidant. Reductants are things that usually have an H in them. H stands for hydrogen. And oxidants usually have no H's in them, and they often have an O for oxygen in them. And so this is a form of chemical energy harvesting, and we do it constantly to stay alive. Actually, all organisms on the planet have to harvest chemical energy in this way. Even the ones who do photosynthesis have to combine reductants with oxidants. So here's some microorganisms. Here's just another example of a microorganism that combines a reductant and an oxidant. In this case, the reaction is your reductant is acetate. You can think of that as vinegar. And sulfate here, you'll see, has an oxygen in it. And if you've ever smelled a rotten egg smell, that's because when these two things react together, they produce hydrogen sulfide gas. So if you smelled rotten eggs at a swamp or a peat bog, or maybe standing next to somebody who had gas, this is the reaction that's responsible for that. Okay. All right, so Earth life needs liquid water, needs to have those building block ingredients, needs to have an energy source. There's one more thing it needs. It needs these conditions to persist. So when we're looking around the solar system, we need to think, are those conditions lasting over time? You know, is there liquid water lasting? Are the ingredients available? Or is there a constant supply of energy from chemicals mixing? Okay, so what about early Mars? Well, a lot of scientists think that early Mars looked a lot like the early Earth. It maybe had some oceans on the surface. Again, those space rocks were seeding everything, bombarding surfaces, so the ingredients are there. And if it was enough like Earth and it had these dynamic hydrological cycles, water cycles, then it probably had some energy sources as well. So I give it 3 checks in all the categories. Kind of arbitrary, but modern Mars gets downgraded quite a bit. And it doesn't seem like liquid water is persisting, at least not at the surface. The evidence that exists for liquid water at the surface is pretty transient. It's not hanging around providing a very good environment. I'd say the ingredients are still there because Mars' surface has been being bombarded by these space rocks that carry all sorts of ingredients for the history of the solar system. Then the energy source, kind of a little more questionable. There may be energy sources. It's not a very geologically active, so there's not a lot of chemical mixing. So that limits the energy sources a little bit. But without liquid water at the surface, energy source really isn't going to get you anywhere for life as we know it anyway. So what's the hope for Mars? The hope for Mars is that beyond the transient water that can form in the surface or the shallow subsurface, there's deeper pockets of water that would actually be more stable because they're under pressure and they could be very salty. But those are going to be hard to get to. It would cost a lot of money to drill down to access these more stable aquifers. There's a little bit of evidence for them, but it's pretty hotly contested at the moment. So I wouldn't say we have proof of these subsurface aquifers, but we're talking something like hundreds of meters or yards. That's a more familiar unit. These aquifers could exist. Now we have rovers on Mars right now that are looking around. This is the Perseverance rover. It's taking samples. It's drilling down on the centimeter scale. It's not getting to these tens or hundreds of meters that I would put money on we need to find an active ecosystem. But it may find evidence of life, extinct life at the surface, something that was once living maybe millions or billions of years ago. It may find evidence. And what's very exciting is those little mini drill cores are going to be sent back to Earth on a sample return mission. So we'll really get to interrogate them in our Earth-based labs. Hopefully we'll find something there. If we do, I'm sure it'll be very hotly contested as to how ambiguous the evidence is as a signal for life or not. Now, if we win the lottery at Mars, in my opinion, we're going to find something like this. Now, this is a picture from Iceland of a fumarole. Essentially, it's a lot of steam coming out of the ground. This could be created by something like on Mars, if you had subsurface magma near a subsurface aquifer, and that lasted for a long time, you could get steam coming out of the surface of Mars. And if our rover happened upon that, that steam could be condensing near the surface and providing a stable habitat for some time. I think of that as winning the lottery because we haven't seen any signs of that yet on Mars, but could happen. Could be very exciting. So Mars isn't too friendly about giving us access to those stable liquid water environments that could exist in its subsurface. Enceladus, on the other hand, Enceladus is a moon of Saturn, and it's giving out free samples of its subsurface liquid water. What do I mean by that? Well, if you zoom in on the south pole of Enceladus, there's these plumes of material being ejected from a subsurface ocean. It's below kilometers of ice, but it's being spewed into space. So it's coming out as water gas and water ice with all sorts of interesting chemicals as well. But there's strong evidence now that it's coming from a subsurface ocean. NASA flew a spacecraft through these plumes of material and sampled them. And the evidence is pretty good that that they're coming from a subsurface liquid water ocean. It's got the ingredients, the CHNOPS, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. They have measured chemical species that indicate that there is energy to be harvested. So this reaction has carbon dioxide plus hydrogen. Carbon dioxide is a very weak oxidant, but hydrogen is a very powerful reductant. So organisms can use this reaction to produce CH4, which is methane and water, and they can harvest energy from that reaction. What's exciting about this reaction is it's thought to be a very ancient metabolism on Earth, possibly one of the most ancient ways organisms harvested chemical energy from their environment. So we have a microbial energy source, it seems, at Enceladus. And lastly, when they flew through these plumes, they found evidence that the place is geothermally active, meaning the water in this freezing cold ocean is cycling down into the rocky interior, getting heated and brought back out and eventually expelled through these plumes. If there is that kind of geothermal activity, that means chemical mixing. That's really good for generating energy sources. So could there be something like this at the bottom of Enceladus? Well, maybe. This kind of energy source that we've shown here, remember how I mentioned CO2 is a weak oxidant? That means it could support single-celled organisms, but researchers think that you need a very strong oxidant to support multicellular and complex life. So you wouldn't see all these animals that are hanging on down here, maybe on the bottom of Enceladus, but maybe single-celled. Maybe we only have evidence right now that there could be single-celled organisms hosted at the bottom of Enceladus. Okay, with that in mind, let's jump to the next place. Oh, sorry, I got to show this cool image that the engineers are working on some very cool concepts to send to Enceladus to explore these fractures in the ice where these jets of material are coming out of. So they're working on a lander concept, and they're working on this snake-looking concept they call EELS. I can't remember the acronym, but the idea is it's supposed to kind of spiral down into these cracks, find some liquid water, maybe in a brine, sample it, and come back out and tell us something about it. This is probably decades off if it happens, but I just thought it was too cool of a picture not to post. Okay, now let's hop over to Jupiter's moon, Europa. And I would say we know much less about Europa. We haven't flown a spacecraft there, specifically for the purpose of hanging out at Europa and sniffing around. But it's got some very interesting features. Like Enceladus, it's thought to have a subsurface ocean underneath kilometers of ice. There are models that suggest there could be enough heat to produce interesting features on the seafloor, like we were looking at pictures of just a moment ago. But we're going to have to go check it out to really know for sure. There's a little bit of evidence for plumes, so material being ejected into space, but you can see the resolution on these images is pretty low. It's hasn't convinced everyone in the scientific community, let's put it that way, that there's plumes. But if they are there, they're probably much weaker than the ones coming out of Enceladus, and they're probably more transient. So they're on and off, maybe. We'll see. We have to go there and find out. But wouldn't it be great if Europa were giving out free samples too? One really excellent thing about Europa is that it's being bombarded by plasma and radiation at the surface. Oh, that sounds bad. Why is he saying it's good? Well, this process, when the ice gets bombarded, it reacts to form strong oxidants, even O2, right? The stuff we breathe, which we know can allow multicellular and complex life, at least on Earth. So scientists also think that this ice is cycled. It looks kind of young at the surface, meaning it could be recycled into the ocean. That would mean these oxidants get mixed into the ocean and are brought to the ocean. This is good. This is chemical mixing, a source of energy. So if we have oxidants being mixed into the ocean, then that means perhaps at Europa, we could be supporting more complex life at the seafloor. I just did a back of the envelope calculation here, but some of the higher estimates of oxygen delivery suggests that Europa could provide enough energy to host hundreds of millions of small fish. I thought that was kind of a cool thought to leave us with. So 7 years in the future, I'm part of this mission team. We're going to be sending an orbiter to Europa to sniff around, hopefully learn some of the mysteries that I touched on. In 20 years, there might be a lander, somewhere around 20 years. Takes a long time to get to these moons, but I think it'll be worth it. And that's it. I'll just say thank you. Here's some images of those missions. These engineers who build these things are just amazing and get them to work. So thank you for your time. And I think we're going to turn it over to Paul.
Yeah, that's right. I love your slides. Fantastic. And I've just got a quick question for you before I say my bit. What's the brown stuff there on the surface of Europa?
So my understanding, the latest on that is, well, they started by thinking out, ooh, this could be some organic carbon stuff. And they were really excited about that. But I think the latest is that it's thought to be irradiated salts. So that might be less exciting to some, but that's my understanding. But I think I would say, that could still change.
Right. Be exciting if some sort of even prebiotic stuff was oozing to the surface, but we'll have to wait and see.
Mm-hmm.
Yeah, I also have one quick comment. You mentioned the polymerase chain reaction. That's the same PCR that we use in COVID tests, right?
Yeah, that's right. So it's
That's
worth
what people
thinking
will be
about
familiar
where would we have been in the pandemic if we didn't have that technology?
Right.
I wanted to make a couple of general points before something specific. So one general point that is always worth remembering is that in connection with Mars, that every time NASA applies a mission to Mars, the media presents it as they're going there to look for life. But the one thing they're not doing is biology experiments. NASA did send a mission to Mars in 1976, and it sampled, there were two lander craft called Viking, sampled the Martian soil and did some tests which were a bit inconclusive, but at least one of those tests, the so-called labeled release experiment, was positive. And it's remained, as one of these incomplete stories over many, many decades. And I personally think it's totally tragic that the Viking experiments can't be repeated in an improved way. And NASA is not interested in doing it. Maybe another space agency will. So we have yet to do biology on the Martian surface since 1976, just worth mentioning. The other thing is, of course, it's a very familiar argument that life got going on Earth pretty quickly. I think Carl Sagan said life must be an easy affair because no sooner was Earth ready for it than up it popped. And it's certainly true that the rapid appearance of life on Earth is consistent with an easy origin. But if you imagine we could cruise around the galaxy and pick a planet at random and find that indeed life had arisen in 100 million years or something like that of its formation, you would certainly be able to conclude that the transition from non-life to life was a very probable affair. Unfortunately, we haven't picked Earth at random. We picked it because we're living on it. Sometimes in science, we call that post-selection. And when you take into account this post-selection, then there's a curious symmetry that the rapid appearance of life on Earth is consistent with a rapid origin. It's also consistent with an extremely unlikely origin, that is where the expectation time for life to emerge from non-life is very much longer than the age of the universe. And that argument can be made statistically precise, and was by the astrophysicist Brandon Carter in a famous lecture at the Royal Society in London in 1981. And I like to mention it because I was actually at that lecture. I was in the audience, and it was notable for being mind-numbingly boring in the way Brandon presented it. But the argument it can be stated informally that if life hadn't got going pretty quickly, then it would not have evolved to the point of intelligent beings like ourselves asking this question, before the window of opportunity closed, because in about another 800 million years, our planet will be too hot for life. So there's a finite window of opportunity, about 4 billion years. And if you want to get to a post-selecting being like us, then that's not really consistent with life starting up towards the end of that window. So we're left in this sort of curious, with a statistic of 1, we're left in this curious situation that it could go either way. So we really desperately do need a second sample of life. And that just brings me to the other point that I wanted to make, because the best hope of finding this second sample is, of course, within the solar system. We know there are many, many extrasolar planets, but it's going to be very difficult just on the basis of observing the atmospheres to be able to say that they actually have life on them. But right here in the solar system, we could grab some rocks and bring them back to Earth in the way you described, Kirt, and then remember the chance of saying, yes, we have a second sample of life. But this is where things again get a little bit complicated. My favorite remains Mars. I've always liked the idea of going back to Mars, doing the Viking experiments again and so on. But the big problem about Mars is that Earth and Mars are not quarantined from each other. And the reason for that is because the planets, I think everybody knows, take a hit from time to time from comets and asteroids. And some of these bigger impacts can splatter rocks all around the solar system. And I always like to tell people that if they come to ASU, take the trouble to go over to the meteorite collection over in ISTB4, they can, if they ask politely, can see Mars meteorites, that is, chunks of Mars that have come to Earth and been collected as meteorites. And when I first got interested in this subject, nobody really knew about these Mars meteorites. There were some mystery meteorites, couldn't quite figure out where they came from. We now know that some of these come from Mars. So rocks travel between Earth and Mars. And your first thought is, well, the conditions in outer space are very harsh. Could a microbe survive a journey on a rock coming, say, from Mars to Earth or Earth to Mars? And it looked pretty desperate at first. But one of the things that, Kirt, you were explaining is that life on Earth doesn't just exist on the surface. It exists in the deep subsurface, certainly at the depths of the ocean, but also inside the rocks, under our feet and beneath the seabed, down to some undetermined depth. The ocean drilling program finds microbes down, a kilometer or so beneath the surface of the seabed. And on land, they can go even deeper. We don't know what the limits are of this deep, hot biosphere. But given that life can live inside rocks, and these rocks can go flying around the solar system, then cocooned in a rock, microorganisms would be screened from the worst conditions in space. And when you take a careful look at this, and people have, you see that there's actually a fairly good chance that a rock going from Earth to Mars would deliver Earth organisms to Mars and vice versa. And because, as you've explained, Kirt, life was warm and Mars was warm and wet 3.5 billion years ago, when we know there was life on Earth, this exchange of material between those two planets would have thoroughly mixed up in any sort of earlier biology. So I have this thought that we might go to Mars, we might drill down into those subsurface aquifers and discover, yes, Mars does indeed have microbial life down there, but it's just borrowing old Earth life. It either got there from here or here from there. And ruling that out is critically important to answering the big question I think everybody wants to ask: are we alone in the universe? Because just finding life somewhere else doesn't mean that the universe is teeming with it if it just spreads around a bit within a planetary system. And that's why I'm more excited, although I think Mars is our best bet and it's easy to get to, I'm more excited by your concentration on Europa and Enceladus, because it's true that in principle Earth rocks can fly out and hit those bodies, but it's a numbers game. I think it's far less likely that these icy moons have been contaminated with terrestrial microorganisms. Although, of course, it's always worth pointing out another little grumble I think we should all have, is that NASA and I think other space agencies are really not very good at sterilizing spacecraft. And so, for example, most of the landers that have gone to Mars have conveyed Earth microorganisms on the landers themselves, never mind any rocks that might have gone there. So, of course, we do have to be mindful of the fact that we could mess up our chances further out in the solar system by not thoroughly sterilizing the spacecraft. I'm not sure there's a great deal else I want to say. I've sort of gabbled through a lot of things. I do think there is one last sort of philosophical point that you explain, Kirt, how very, very carefully how the building blocks of life are ubiquitous, and we find them, for example, amino acids anyway in carbonaceous chondrite meteorites, and it's true that those building blocks are probably spread throughout the universe. But I think there's a huge difference in going from the building blocks to the building, just as there would be if you said, well, there's no shortage of bricks, so there's no real mystery about where New York came from. It's, you've got to put the building blocks together. And from my own point of view, I think the big unknown about going from non-life to life is not so much the hardware. That's your job, you're a chemist, so you worry about the chemical stuff. But also we have to think about the software, about the informational content of living organisms, and how that information management and encryption in the genetic code, how that came into existence. And I think that's still a deep mystery. So I'm like you, I think I sort of have an optimistic bent. I would like to feel that there is a principle, a deep life principle in nature, a work in the universe that means life will emerge, and that its information, the software and the hardware, will somehow emerge wherever conditions permit. But as a scientist, I have to admit that as we don't know all of the many steps between the building blocks and the simplest living thing, we must be open to the fact that it could have required just a dream run of extremely improbable processes that happened here on Earth and we're the product of it, or at the other extreme, it could be something which happens everywhere. So that gives added urgency, I think, to astrobiology, to actually taking a look, because I don't think any amount of armchair theorizing is going to give us the answer as to how likely it is that non-life is going to turn into life, and how long it will take on average. And I think that's one of the questions that came in whilst you were talking. So I think I've said enough, and I think hope Maulik can feed us some questions so we can develop the discussion further.
Thanks, Paul, and thanks, Kirt. And let me remind everybody that if you have any questions, now's the time to enter them into the chat. We already have a few. Let me begin with a question of my own. I guess this is for Kirt. Sometimes people have speculated that there would be other forms of life that are not carbon-based but based on silicon, or that there might even be other aqueous solutions that are alternatives to water. Just from a chemistry point of view, how likely are these, and what is the role exactly that carbon, and what's so special about carbon and water?
That's a great question. It's also a really tough question. Before I take it, should I unshare my screen or should I keep it up?
Maybe you should unshare it. Nice though your slides are.
Yeah, I think there are some, well, it's a really tough question, because for the conditions in our solar system and the conditions on the planets here, carbon does seem to be special. It has the right chemistry to have lots of bonds with different types of atoms, which gives it a lot of functionality. And it has the right kind of strength of its bonds under our planetary and solar system conditions, that they're They're just hard enough to break that it doesn't fall apart, but they're just easy enough to break that you can play with them like Legos and build cool things. That's what life does. So I do think carbon is special, at least for our solar system. I think water is also a special solvent in our solar system because of the way it interacts with our biomolecules. Of course, we're being very biased here. The fact that water has a lot of interesting properties. One really good one for on the planetary scale is it's one of the few liquids where its solid form, ice, will float. And this has huge planetary implications. If like most materials, when you do the liquid to solid transition, it becomes more dense. Let's say, metal, for example, when you take a metal liquid and it cools down and solidifies, it's more dense, it sinks to the bottom and the liquid metal would stay on top. If water had that property, it would cover the seafloor in ice, and there wouldn't be this chemistry that happens when water permeates down into the rock, gets heated, reacts with the rock, and brings all these nutrients back up. That kind of process, we don't know, but it could have been totally crucial for the origin of life on Earth. So yeah, there are special properties, I think you could say, but around a different star with totally different planetary conditions, maybe there are other combinations of ingredients that work. I can't really speculate beyond just telling you a couple cool facts about what we're made out of, though.
All right, thanks very much. That was great. So there are a couple of questions. I think I directed Paul. This is from Howard Lannis and also Gerald Orozco ask essentially. You mentioned that microbes could have made it to Mars from here on contaminated spacecraft. Wouldn't the cosmic radiation kill most terrestrial microbes on a spacecraft?
Yes, the answer is most, but not all. And I think the worst thing to get you into planetary space is really not cosmic rays, it's solar ultraviolet radiation. That really is deadly to almost all known organisms. But if you're shaded from that, and this seems to have been the case with some microbes that have been recovered from the moon, that were on the spacecraft. If you're shaded from that UV, then there's not a problem really about the vacuum and the low temperature of space, because many microbes love to be freeze-dried, and on arrival you warm them up and they start up again. And so it's I'm not saying that it's a comfortable ride, but it certainly is the case that if you wanted to really thoroughly sterilize a spacecraft, you'd have to spend a lot more money than is being spent at the moment.
Thanks. Now there's some questions about other options besides Enceladus and Europa. Peter Klupar and Daniel Gulick asks about Titan and also the clouds of Venus. So Kirt, maybe you want to.
Sure, I'll take Titan first. The surface of Titan is much, much different than anything we're seeing in the solar system. It has hydrocarbon lakes, lakes of, I think they're nitrogen, well, propane, methane, ethane, butane. And so if there was life on the surface of Titan, it would be very, very different than Earth life, very, very wildly different, because it's a different solvent. It wouldn't be water that it's living in, it would be something else. I find that unlikely just from a chemistry perspective, thinking about what we know about how chemicals behave in these solutions. But I wouldn't rule it out until we go there and have a look. And there are missions coming up to do so. The thing that makes Titan, I think Titan is a very interesting place to go and explore for lots of good chemistry reasons. I'm a little less excited about the prospects of finding an active ecosystem there, because the ice on Titan, I believe, is much, much thicker and doesn't, our observations of the surface suggest it's older and it doesn't recycle as well. So we may not get, if it does, it is thought to have a subsurface ocean, but if there's an active ecosystem down there, we may not get the signal back to the surface. And I don't think we're going to be able to get through all that ice on the budget that NASA has. The other one was Venus. Maybe you can speak to this, Paul. I just have one thing to say about it, and that is on Earth, we're just starting to scratch the surface on finding microbes in the atmosphere. Well, that's work that's they've been observed, but whether they can have a full life cycle in the atmosphere or whether they need to have part of their life cycle on the ground and then they just get transported through the atmosphere and then land somewhere else and have more of their life cycle, that's still debated, unknown, I think you could say. So that's about the extent I can comment on life in the clouds. I think we don't really know if it can, with all this dispersion and chaotic behavior that happens in an atmosphere, I don't know if that's a stable enough place for a life cycle of a cell.
But I think that's a great point, Kirt. It's exciting to think we've got a deep biosphere that life extends down into the Earth's crust, but it also extends up into the air, and we don't know how far up. The higher you go, the more UV you're subjected to. So again, it's not a very pleasant environment, but we know that that some microorganisms can be remarkably radiation tolerant, and it could be that that there is a transport mechanism that they go up, they come down. I think there's a lot to the biosphere in the sky. It's a lot to be explored. Whether you could do that on Venus, there was a flurry of excitement about a chemical called phosphine that I confess I'd never heard of, but I guess, Kirt, that's the sort of thing that you learned when you were at high school. But phosphine was supposed by some to be a possible signature of life in the Venusian clouds. We should point out that Venus is a horrible place down on the surface. It's immense atmospheric pressure, temperature that will melt lead, that you don't want to be there. But up in the clouds, it's possible. But I think this phosphine signature either went away or is no longer considered very significant. So I've gone off the idea of life in the clouds of Venus.
Okay, we have a question from Barbara Temple, and this was sent by email. She asks, is current technology up to the task of spotting biosignatures reliably? So, Kirt, you mentioned that you want to go and check out these planets. So what would we look for, and could we do it?
Well, I think Paul made a really good point earlier when he spoke that there really hasn't been a good experiment sent in many decades to look for active life. Signatures, most signatures are going to be hotly contested because the signatures that life produces on Earth, 20 years ago, someone thinks something's a biosignature, but then some progress happens in chemistry and you realize, oh, you can create that signature with non-living chemistry if you create the right recipe. There are, scientists have developed some scales where they say what's a weaker signature, what's a stronger signature. One of the stronger signatures that I really like is it's called chirality, and it's the handedness of a molecule. So if you have two molecules, some of them have a certain 3D structure to where they can be identical, except that they're the opposite handedness. You can't superimpose my two hands, and you can't superimpose those two molecules. Well, it turns out all the amino acids, that string of pearls I was talking about, the amino acids that make up the protein in sort of a string of pearls, in our proteins, they're all left-handed. None of them are right-handed. And so a strong biosignature is if you find an abundance of amino acids that are one-handed. And by abundance, I mean nearly pure. You would need it to be a strong biosignature. You wouldn't want any of those right-handed amino acids mixed in. But even better than that would be if you found only right-handed amino acids, because on Earth, life only has left-handed. So if you found only right-handed, that would be a strong biosignature for perhaps a second genesis, or at least something that deviated from Earth's evolutionary history early on before this handedness took over. We don't know exactly how early that might have happened. But
can I make a quick follow-up comment? Because on the Viking experiment, Gil Levin, who designed the labeled release experiment, he was a familiar figure in the Beyond Center. He died 2 or 3 years ago, but he would often come and lecture on this topic, and he was convinced he found what it was. But in the original concept, he wanted to have chirality discrimination. So he wanted to send not just a nutrient broth to Mars to pour on the Mars dirt, but a sort of mirror broth, which would consist of a nutrient medium with the opposite chirality molecules. And then if the samples fizzed with one but not the other, that would be much stronger evidence that it was biology. But if it fizzed with both equally, then that would suggest it's chemistry and not biology. And to test that concept, NASA actually had a project in the Atacama Desert, which is very Mars-like soils. And sure enough, in the middle of the Atacama, where everything is sort of desperate and almost nothing lives, you get the soup and the anti-soup fizz equally. And as you go out towards the wetter outer regions, the asymmetry begins to build up. So I think if we ever do repeat Viking, it's got to have that chiral discrimination thrown in. And that's what Gil wanted to do right up to the time he died. But alas, we're still waiting.
But for that, you need actually to get possession of the amino acids, right?
No, the way this labeled release experiment worked was that a nutrient broth was sent on the spacecraft and poured onto the sample of the Mars soil. And then it's called labeled release because it had a radioactive tag that could detect the carbon dioxide given off in the headspace above the sample. And so what you do is you have a little capsule of soup, and then another capsule of anti-soup, the mirror molecules of the same thing. And if life has one chirality, if it's homochiral, doesn't matter if it's the reverse one on Mars, it'll guzzle one, but find the other unpalatable and won't progress with it. So that's the idea. You test it with both and see if it fizzes equally. It certainly fizzes. Nobody denied that Gil's experiment produced carbon dioxide. It fizzed. The question is, was that just some sort of soil chemistry that people have struggled to replicate, or was it actually microorganisms in the surface? A bit desperate for life to still survive in the surface soil of Mars. It is such a harsh environment, but there's life inside. If you go to the Atacama Desert, even in the inner core, there are microorganisms that live inside the salt, pillars of salt that exist there, and eke out a living with photons in the daytime and the H2O deliquescence, where the water invades the salt matrix at night when the dew point the air temperature goes below the dew point. So who knows? It's not impossible, but we might find something yet on the surface of Mars.
So we have just about time for one last question, and you mentioned the inhospitability of Mars, and so I'm going to ask. So Joyce Pirtle asks, well, what do you think it would take to make Mars habitable?
Terraforming, yes.
Terraforming question.
There are whole books written on it, and I think given the time available, the one thing we can say is what it takes is an immense period of time. It's not something that you could do in a few years. We're talking millennia to change anything about the surface of Mars. But people have thought about it.
All right. Well, I think we are just about out of time. So thanks everybody for listening, and we'll see you next month. I don't think the topic has been announced, but
it'll be something completely different.
Well, it'll be something different. So thanks for listening and have a great evening. Bye-bye.
Thank you, Maulik.